This episode of Cell and Gene the podcast is brought to you in partnership with Thermo Fisher Scientific and Applied Biosystems' new QualTrack real-time PCR and digital PCR solutions for biopharma. Give your cell and gene therapy development an edge with reliable and accurate qPCR and dPCR workflows backed by a trusted supplier. Explore the complete ecosystem of CGMP-compliant qPCR and dPCR assays, master mixes, and instruments at thermofisher.com/qPCR/biopharma. Hello, listeners, and welcome to this episode of Cell and Gene the podcast. I'm your host, Erin Harris, and my guest for this episode is Will Junker. He is the head of Vector Manufacturing Quality at Kite Pharma. Will, welcome to Cell and Gene the podcast. Thanks for being here with me today.
Oh, thanks for having me, Erin.
Absolutely. All right, we're going to jump right in. So talk to me about what you feel from your professional experience. What are the biggest challenges facing viral vector manufacturing today? And on top of that, do you see any real change coming soon to alleviate the challenges that we're going to go through?
Yeah. So certainly the largest challenge that we're facing at this point in time is the limited CMO capacity for plasmids, lentiviral, AAV, as well as retrovirus. And what we do, or the reason for that, is it is still a niche commodity compared to large-scale biotech manufacturing. In the light of cell and gene being still even though being an industry success story, right? So we have capacities established to at the CMO and CDMO community that supplies some, but certainly not long-range commercially enough vector to satisfy the cell and gene therapy market. So we have a lot of industry responses around this, right? So it starts with multiple new companies coming, I would say, out of the woodwork, right? In general, companies that have been providing like equipment or technologies to the cell and gene therapy space, and have identified this as a strategic target and a need across the industry to have that capability to manufacture vectors, and work with their client and sponsor community to building this capacity. Some of them already have capacity established, not just the classic CMOs we used to work with, a couple of years ago when this was not really recognized as a bottleneck. Kite's approach, we do vertically integrate. So we are at a point of our commercialization journey where we have a couple of commercial products, a couple of late-stage pipeline products, and we understand what our vector demand is going to look like over the foreseeable future, up to 10, 15 years in the future. And what we are doing is, or what we did is, we built our own capacity for viral vectors specifically. I'm running quality at this facility, and we are ready to supply the internal demand certainly over the next, over this foreseeable future, with all kinds of vectors, right? And this is, I think, commensurate with the maturity of our company.
Gotcha. Okay, that's good. And we're going to get to specifically capacity a little bit later in our discussion, so we will revisit that. But in the meantime, I want to talk a little bit about, standardizing or developing standardized CMC methods. It seems to be a bit of a challenge for the industry as a whole. And so I wanted to get your take on, why standardized CMC methods are so important, what those methods are, and, if and when developing, is it even feasible to develop standardized methods, standardized CMC methods?
Yeah, that's a really good question. And it's, you're totally right. You're spot on. This is a challenge for everybody in this field. So in general, to look at cell and gene therapy, all of our our entire field, our industry, all the providers of these therapies struggle with the more exotic and novel nature of our analytical methods across the field. We look at things like impurity profiles, potency, safety methods, whatever that looks like in the analytical field, in a way that we try to lean towards best practices, but a lot of this is internally developed. And if you talk to one analytical development department at one company and another one at another company, you will very soon find significant, profound differences in the way this is approached. This is not helpful if you go commercialization and you go to your second product into a pivotal phase 2 or 3 study, and you have to reinvent the wheel, how to manufacture and how to control manufacturing of a consistent quality vector. So the trick is really to standardize, to come to first of all, to come to platform methods, right? This is the long-range target, to have a certain assay that is being generally accepted across the industry, has the positive impression with the health authorities that regulate our industry, and then that is also reproducibly able to be transferred in a way that that makes it easy, right, for QC or analytical development departments to adapt them. So so that's what we're trying to do. We're working with ISO and other institutions out on the market as an industry forum, if you want, right, to like streamline, even just start developing of such standards and associated platform methods.
Good. Okay. In keeping with the topic of standardization, with biopharma companies using different production systems and downstream processes. So talk to us a little bit about, in your experience, is this good? Is this bad? Is this indifferent? And perhaps does it matter to, a Kite Pharma as in the same way that it matters to, a small and emerging biotech? Can you talk us through that a little bit?
Yeah. This is another one of the things that sometimes keeps me awake at night, and together with our leadership team. So it's an interesting commodity to look at a vector. It is breaking apart with the original upstream downstream model that we have adapted to across the industry, coming from monoclonal antibodies, come from general regenerative commodities. So vectors are tricky to standardize in their manufacturing process because they are, they vary in their payload, meaning the package, proprietary information that the success of every cell and gene therapy company is based on. So we can standardize manufacturing of types of vector. We have one way to make retroviral vector at Kite, and this is a general way of working across the industry. When it comes to lentivirus and adeno-associated ones, or even new ways of introducing these payloads, right? It becomes a little bit more tricky to look at, okay, what is the type of bioreactor? What's the scale you want to manufacture these in? Do you do adherent or suspension cell culture? What does purification look like? Every one of those vectors may have a different specific ask when it comes to impurity profile, what's acceptable and what's not. So there are different ways of doing this. So I think it's generally a good thing that we haven't gotten to a standardization there along the lines of what happened in large regenerative biotech. But we're still struggling with this, right? We don't really know yet how the market develops. At some point in time, you can maybe buy an off-the-shelf, equipment package that is easily put into an envelope where you can do aseptic manufacturing of a viral vector, in your garage. but I think we're still a couple of decades away from that, status.
Gotcha. Along the lines of what I thought you probably were going to say, that we're a little ways out, but makes sense. I want to go back to capacity, manufacturing capacity. What are some of the key issues from your perspective when it comes to capacity planning? And how do you and your team at Kite, or if not, perhaps you and your team at Kite, just from your professional experience, how do you address those key issues?
Yeah, capacity planning is really difficult. So I want to align this a little bit with the cell and gene therapy journey of any one of the companies out there that is, embarking on it or already on their way. So at the beginning, you find the science to really have a breakthrough therapy, at your hands. Your best guess is that it's going to work. You go into clinic, you may have in phase 1, some successful trials, and you find out, okay, this is really going to be awesome to add to the market, right? It may meet an unmet need, or it has really a great potential to make a difference. So the trick is to work with the CMO or CDMO at the beginning, because you don't really know what your capacity needs to be, due to the fact that you have very little or limited understanding of market penetration and generally the success of your cell and gene therapy. Working with those CMOs was for us very effective. We were able to have sufficient supply to not have any shortage even over the pandemic last couple of years. But we outgrew that. So we are right now looking at this vertical integration of vector manufacturing into cell and gene therapy, or specifically CAR-T at Kite. And we have made great strides and great investments to establish that, even investments with our CDMO community to soften the blow a little bit. And so where we are is we are at the stage where we have a long-range plan for market saturation, a strategic long-range plan to really understand, what is our capacity increase for CAR-T, or for cell and gene therapy, which then in turn drives that capacity for viral vector. And we have established sufficient capacity to keep us not just afloat, but actually well established over the next couple of decades.
Good. Okay. I want to talk about, from a time perspective. So especially as it applies to cell and gene therapies, time is of the essence for every facet of it. Everything is a race against, like I always like to say, a highly regulated clock to bring these therapies to market, and to patients. Talk to us about time as it applies to viral vector manufacturing. So in other words, when should biotechs start considering their manufacturing processes? Should they look to outsource? And I know we just kind of talked about this. Okay, outsource early, work with a CDMO. In what ways can listeners make time their friend when it comes to viral vector manufacturing?
Yeah. So yeah, first of all, it is extremely beneficial to work with a CDMO, due to the fact that there's expertise that you buy with the contract, right? To establish capacities in a way that helps you get up and running in your cell and gene therapy journey. When it comes to the actual provision, there is a beautiful contrast, right? So cell and gene therapy in general, as you said, is a time crunching type of activity, right? You have a time to patient commitment, generally as a company. You have to deal with things that are GMP related, like I'm a quality guy, so there are deviations, there are kappas, there are issues with closing a batch record. There are a myriad of little things that could happen. You might have a visual inspection failure or an out of specification in a release assay or something like that. So it's always a time crunch, and on everybody who is working on the manufacturing process themselves of cell and gene therapy knows that their main goal in life is to make those and to help that patient. When it comes to, we are in cancer, specifically hematologic malignancies. Our patients, at least at the beginning when we were, kind of end of line in our positioning, our patients had like maybe just enough time to wait for dose to be made and for infusion before, they were thinking about retirement. And so what we need to do is at that case, really be consistent and be fast. Speeds up the essence. For viral vector, the contrast that, for viral vector, the manufacturing capacity utilization is really a question of inventory. Mm-hmm. So much more along the lines of traditional biopharma, we are making enough vector to supply our network of T cell facilities, for CAR T to be able to always meet the demand of the market. And the demand is a moving target, right? We're expanding demand utilization, at a certain velocity. There are new things that pop up, new programs, adjustments, process improvements that require, a sometimes massive change to our vector manufacturing, model. And so, so the thing is, focus on inventory when you make vector, and don't necessarily worry too much about time to market unless you have issues with expiry of individual vector lots, getting too close to consumption, where you need to make some adjustments, either file for expiry extension or improve your process.
Gotcha. Okay. And a concentration on inventory is a nice segue into our next topic here, which is to help our listeners prepare for 2023 and beyond, and to really consider the absolute top priorities when it comes to vector manufacturing quality. But, inventory certainly being, one, having a big focus on that. What else would you say would be the absolute top priorities for anybody listening for the foreseeable future?
Yeah, you nailed it, Erin, right? So the difficulties that we faced during the pandemic was supply shortage in general, and specifically to vector. And a couple of the commercial players had issues being close to or beyond stock out of therapies, just due to the fact that they couldn't get enough vector to supply their capability. So yes, capacity is continuing to be a focal point of any development in this space. But the other thing is also process understanding. So from a quality perspective, we see oftentimes vector manufacturing processes, and that is not Kite specific, this is across the industry, being kind of a not an afterthought, but a side gig. With the scientific, inclusion of whatever cell and gene therapy into a commercial field. And so to understand, however, the vector manufacturing process at a higher level than we do today, going, a little bit and actually significantly above just understanding the science and looking at this attractive technology with its great efficiency to make a vector that really works. We had very interesting conversations with our health authorities, with the FDA, the Europeans, et cetera, to around what is our process understanding really? How do we control impurities? How do we control multiple vectors being manufactured in the same building? And how do we what's our future projection on how we continuously and securely supply the market, within Kite to be able to continue to make therapies. So process understanding is so important. And then, it's always important to have a good understanding of the pathway forward, not just, hey, we're done, have a check mark on vector and start going into the clinic. That's not the case. Even working with the CMO or CDMO, accountability for the quality and the continuous supply of vector lies with the sponsor company. And so, that's not a reason to kind of just, walk away from it.
Yeah, sure. That makes a lot of sense. We've kind of formally come to the formal end of our discussion. And
Okay.
So I wanted to ask you to get to know you a little bit better. And correct me if I'm wrong, you are a Californian by way of Germany. And
that is true, yeah.
That is true. And as the summer winds down, even in California, I was wondering, what do you like to do in the quote unquote fall months in California?
Yeah, that's a good question. So, California, even the winter temperatures during the day in Southern California are still in the 70s, close to the ocean. I live just a couple of miles away from the beach. So I'll continue to go to the beach, not spending too much time or not that much time anymore in the water, but rather looking at things like, paddleboarding, surfing to some extent, but certainly taking long walks. I will take my son hiking, and we're going to have a good time on this. I'm going to spend a lot of time with my girlfriend preparing for Halloween. Halloween is our favorite holiday. So this is always a good time to let the scary out a little bit if you want, right? And then of course there's Thanksgiving, there's football, there's the general American pastime when it comes to the holiday season.
That's wonderful. That's so much fun. It sounds like you have a full lineup.
Oh yeah.
Do you dress up for Halloween?
Oh, most definitely. Yeah, I'm trying to be
Really?
the scariest person on the block.
Well, you'll have to send us some pictures after the fact, and we can post them on Cell and Gene for our listeners and readers to see exactly what you went with. Do you know what you're being?
No, I'm still
figuring it out. That's great. That's great. All right, listeners. Well, that wraps up this episode of Cell and Gene, the podcast with Kite Pharma's Will Junker. Will, thank you so much for all the valuable information you shared today. This was a pleasure.
Thank you, Erin.
Be sure to visit Cell and Gene to register for our free newsletter and to get all the quality content you need to help you perform your role even better. Talk to you soon.